Welcome to Crossroads. I'm Jerry Kay, journalist in residence for the AP Giannini Foundation. I recently met with Dr. Asmin Tulpule, an AP Giannini postdoctoral fellow, to learn about his medical research. I first asked him to tell us about the career path that led him to study pediatric cancer.
I work in two worlds that are 100 steps apart on the campus of the University of California in San Francisco. In one world, I am a pediatric oncologist, which means I take care of children with cancer. I am part of a large team that diagnoses, recommends treatment plans, administers chemotherapy, celebrates with my families when their cancer is gone for good, because in pediatrics, we get to say that more often than not. But I'm also the one who has to tell families when their medicines are failing, who grieves and cries with families, and tries to help parents cope with the unimaginable sorrow of having to lose a child. This is a sobering and humbling experience, and it is a true privilege to be able to care for these families. In my other world, 100 steps away from the Children's Hospital, I just started my own independent basic science research laboratory, the Tulpule lab. We study human cancer in the lab. We wear lab coats and sit at benches and grow cells in dishes and add drugs. We are working for cancer, searching for the next cures. But there isn't any of the raw emotion of the clinic. Science is a very analytic endeavor, and the lab is a pretty happy and communal place filled with pop music. The disconnect for me is tangible. There are days when I walk back and forth from clinic to lab, transitioning from physician to scientist and back in the course of minutes. And really, I'm always both.
The two worlds you describe seem quite separate. Some might ask, why not just focus on being a doctor or a scientist? Why both?
It's a good question, because the training to become a doctor and the training to become a scientist are both long and non-overlapping. But I've always been a scientist first and foremost. Both of my parents are PhD scientists, I grew up in a household of science, and I've always had this fundamental curiosity to understand how things work. I went to graduate school fully intending to become an academic scientist. But it was there where I started studying human diseases, and I began to meet families whose children were affected by the particular protein that I was studying in the laboratory. And it was there I found that I really enjoyed this human side of medicine. I loved taking care of families, meeting them, and getting to know their stories. But it's also this human side that pushes me back into the laboratory, because every time I step into the clinic, I face the reality that cancer is scary, that cancer kills, and our medicines, even though they're getting better, are still not nearly good enough.
What do you study in the laboratory?
So I study a bone cancer called Ewingssarcoma. The word sarcoma means cancer of the bone and soft tissues, and this is an area in pediatric oncology where we are in desperate need of better therapies. From a scientific standpoint, Ewingssarcoma is very interesting because we understand a lot about the disease. We know exactly what causes it, which is one and only one genetic change in a single cell that creates a cancer-causing protein called EWS-FLI1. And we've known about EWS-FLI1 for 30 years, but the problem is that no one has been able to design a drug to block the cancer-causing protein, EWS-FLI1. My research takes an alternate approach. It says we know EWS-FLI1 gives the cancer cells great strength, but perhaps it also leaves behind one tiny weakness that if we could find, we could use to selectively kill cancer cells. And that's important because right now, if a young man walks into my clinic with a painful lump in his arm, and we take a biopsy and we find out that he has Ewingssarcoma, the treatments that I have to offer him are 5 different toxic chemotherapies that we've been giving to patients since the 1980s. And while we are able to cure some of our patients, our treatments continue to fail far too often. And even for those fortunate survivors, they're saddled with significant long-term side effects from their therapy, including heart failure, infertility, and second cancers. So that's why I'm in the laboratory, trying to find a better, more tactical way to target Ewingssarcoma.
So, what have you found?
I became interested in a very fundamental process that all cells, cancer cells and normal cells, have to perform in order to survive, which is repairing damage to their genetic material, or DNA. And specifically, there is a very lethal form of DNA damage known as a double-strand break that all cells have to repair. And because of this, normal cells have a lot of different backup systems. So I like to use an analogy to explain this. So imagine DNA damage as the rain. If that's the case, then all cells live in Seattle. They are constantly getting bombarded. And so normal cells, they take precautions. They carry an umbrella, they wear a raincoat, they wear rain boots. Now cancer cells can't just ignore the rain, but they don't want to carry all around all this rain gear. They want to be able to live free and move fast. And so while normal cells will often have 3 protections against the rain, cancer cells like Ewingssarcoma will often have just 1. And this is the key point. If you can identify that one piece of rain gear that a specific cancer cell is dependent on, and I believe I've done that with Ewingssarcoma, then you have the opportunity to target. So how does this work in practice? Let's say Ewingssarcoma cells only carry an umbrella. Well, you could then bring in a drug that turns on the wind, flips that umbrella, the Ewing cells get soaked and they die. But now what happens to normal cells? Normal cells will be fine because they have a raincoat, they have boots, and what I just described is an example of a specific, targeted medicine with minimal toxicity to patients that I hope can become a reality so we can better care for our patients.
My thanks to Dr. Asmin Tulpule. If you'd like to learn more about the fellowship program or to watch more of our Crossroads videos featuring other Giannini fellows, please visit our website.